Dear students, a warm regards to all. Today, I'd like to give a brief idea regarding hardening in plant tissue culture. Plant tissue culture is nothing but developing a new organism from a single cell in the lab condition. And this hardening is the last stage of micropropagation or plant tissue culture. And this hardening, it's an adaptation in greenhouse before transferred into the ex vitro conditions are the plants are introduced into the field. So it's a gradual process. Hardening is nothing but introducing the plantlets naturally into the environmental conditions. First, we take the plantlets from the culture medium. These are the plantlets, end product of plant tissue culture formed by the action of auxins and cytokinins from the callus tissue. Callus then differentiated mass of cells, undifferentiated, unorganized mass of cells to be the callus. From the callus, these plantlets are formed. First, we take the plantlets carefully from the culture medium. Then wash away the agar. Actually, plantlets are placed in the agar-agar culture medium at the time of plant tissue culture. So wash away the agar residues. Agar-agar is a gel-like non-nitrogenous substance extracted from the cell wall of rhodophysian members, red algae. So first, we should remove all the residues from the plantlets. Then the plantlets are treated with fungicide. Before it should be planted in a small pot. The fungicide treatment given to the plant. The chemicals used to kill the fungus are called the fungicides. So first we can treat the plantlet with this fungicides. Then the plantlets are transferred into the natural environmental condition is not feasible because it is already in the in vitro condition in a controlled environment the plantlet is grown. If you can straight away transfer the plantlet into the natural environmental condition, it is unable to survive from a protected environment to the natural environmental condition. The survival is somewhat difficult. So after treating with the fungicides, the plantlet is placed in a pot. It's very important, containing soil, the material needed for the growth of the plantlet. Then the plantlet is covered with perforated the plastic bag. This perforation are the small holes in the plastic packet for aeration, for gases exchange and transpiration. This pores, these minute pores are very, very important. So through these pores, aeration takes place. Then these plantlet covered by the plastic bag placed in a shade or low light for 15 to 20 days. Because gradual change in the environmental conditions takes place. Then remove the polythene bags for a few hours every day for better sunlight to perform photosynthetic activity. Then increase this plantlet to the sunlight for more time. Finally, the plantlet is introduced into the natural environmental condition. Here, this poly bag behave like a greenhouse or it give a protective environment. Then these plantlets are introduced into the natural environmental condition, the field, the process, the entire process is said to be a hardening. This simple picture completely explains what is going on during hardening process. The plantlets are gradually introduced into the natural environmental conditions is said to be hardening. Plantlets are taken, washed with water to remove the residues. Then we give fungicide treatment or placed in a pot containing the soil material or growth material. Then it's covered by a plastic bag, a polythene bag with the minute pores in it. To the pores, gases change or takes place or aeration takes place. After 10 to 15 days or 20 days, it is accustomed to the environmental conditions, then it is placed in the field or planted in the field. This entire process is said to be hardening. Thanks a lot for hearing. Thank you very much. Thank you, thank you, thank you, students.